Welcome, everyone, to the Science from the Fringe podcast, first ever in-studio podcast. We're in an undisclosed location. My name is Bryce Nichols. I'm a professor of genetics at Rutgers University and co-founder of Biosafety Now. I am joined today by Neil Harrison, who's also a member of Biosafety Now and a professor at Columbia University. How are you doing today, Neil?
Doing good. Thanks, Bryce.
Neil and I are going to talk about the science of the COVID-19 pandemic. Now, you are one of the lesser known, I'm going to say it, heroes of the COVID origin space. Can you introduce yourself and tell us a little bit about your role in the fight for the truth about the origin of COVID?
Okay. So I'm a professor of pharmacology at Columbia University, and I first became interested in the COVID-19 pandemic before it happened. I became aware from news reports that something was happening in China, and I spent a little bit of time informing people at Columbia that we might experience an epidemic in New York City. I think I was probably one of maybe a dozen people independently at Columbia who were wandering around the building trying to raise the alarm and get people's attention that something bad might happen.
Though, to be fair, before the pandemic, you were found to sort of be wandering around the hallways.
Often.
Right.
Often, which is probably why they didn't pay any attention to me whatsoever. So as events proceeded in Wuhan and in Italy, I became more and more convinced that there would be an epidemic in New York and redoubled my efforts to make contact with the administration so that they would be ready to do something if the virus actually arrived in New York, as it eventually did. After that, I spent considerable time working with my clinical colleagues. I worked on a clinical project where we analyzed blood from COVID-19 patients. We were looking for biomarkers of severe COVID-19 that might be associated with severe disease and death. And we were successful in identifying a couple of factors that had been missed by other studies that were larger studies. And I really didn't think very much about the origins of COVID until later in the year 2020, by which time people were beginning to write a little bit more. There were papers appearing in the literature about the possible start dates for the epidemic in Wuhan. And people were beginning to speculate about which bats the virus had come from in which parts of China and so on. And then there were even some other speculations being put forward about other possible origins that weren't fully natural in nature.
And after that, you published a paper with your colleague, Jeffrey Sachs, in 2022. Can you tell me a little bit about how that came to be?
Right. So I guess in 2021, I was reading a lot of the things that had been published, and I became somewhat skeptical of some of the official stories, and I was quite interested in other possibilities. For example, we knew that there were groups that went out to collect bats and collect material from bats, and I wondered whether perhaps they'd inadvertently brought the virus back to Wuhan, and that could be a source of the original epidemic in Wuhan. And that led me to read a fair amount about the activities of EcoHealth Alliance and become aware of Peter Daszak and some of the other people associated with that group. About the same time, and completely independently, Jeff Sachs was learning about some of the same people because he'd been asked to lead an inquiry by The Lancet. So there was a Lancet Commission on COVID-19. Jeff was asked to lead that. And as part of that effort, there was a group that was convened to look into the origins of COVID-19. And as part of that group, Peter Daszak was recruited. This included many other associates of Peter Daszak. Gerald Koish from Boston University was one of those people. Peter Hotez was involved from Houston. And there was a number of other luminaries in virology, evolutionary biology, and medicine that were recruited as part of that group. As that inquiry proceeded, Jeff Sachs became more and more convinced that they were pulling the wool over his eyes, basically, and that there was a group of people associated with virology that was only interested in pushing one particular origin story, and that other lines of inquiry were essentially being completely closed off. And so at some point, he began to question some of the members of the commission about their potential conflicts of interest. And of course, everybody associated with official virology had a conflict of interest in the sense that they get large amounts of grant support for doing work that is either directly or indirectly related to so-called gain-of-function work in virology. And so he felt that many of the people involved in the commission had conflicts of interest. Peter Daszak had a particularly obvious conflict of interest because of his association with the Wuhan Institute of Virology. At some point, everything came to a head. The commission was temporarily disbanded, and then another group convened itself to produce an alternative report on the origin of COVID that was published separately, apart from the main Lancet Commission report. So I got to know Jeff Sachs a little bit at that time, during the period where he was producing a semi-independent report, and this other report was being produced by a group that was led basically by Gerald Koish and Peter Daszak. So Jeff Sachs and I started writing this, I think, early in 22. We probably started writing in January 22.
Now, hold on. He leaves the Lancet Commission.
Yep.
And you knew him from before, or was this a new, how did he know to work with you on this? Or did you reach out to him?
So I'd written a piece about Peter Daszak, which was less than complimentary. Basically, I felt from much of what I'd read that the whole business of pandemic prevention was, in essence, it was a con, because it wasn't really possible to go out and collect hundreds and thousands of viruses and then automatically be able to figure out that one of those was going to be able to cause a potential pandemic. And basically, I ended up saying things that were very similar to what people in mainstream evolutionary virology had already said. So they basically wrote something expressing their skepticism about the usefulness of going out collecting hundreds of thousands of viruses in the same way that the Victorians went out and collected thousands of butterflies that are now in the Natural History Museum everywhere. Many people have described that kind of science as stamp collecting, and I think it was Ebright that pointed out that this was stamp collecting that could cause a global pandemic by accident, because they could easily collect a virus that was brought back to a lab of which the hazards were unknown and of which they would be completely unaware. So there were a lot of problems in having Peter Daszak in particular looking into the potential origins of a global pandemic.
Didn't you, the title of your article in Medium was what? Do you remember? You called him Peter Daszak is the...
I suggested that he was the Elizabeth Holmes of pandemic prevention.
That's right.
A very accomplished con artist who was able to get funding from many, many famous people to get her startup company, Theranos, underway without any apparent scientific underpinning for the endeavor.
So Jeffrey Sachs reached out to you, or you reached out to him, or how did that?
So I actually wrote something internal, which will remain private, but it was in the university system, and Jeff Sachs basically assented or agreed to what I was writing.
Oh, okay. So then you collaborated to get that out then. Okay.
Right. So we started working on this opinion piece for PNAS. So PNAS is the Proceedings of the National Academy of Sciences, and the National Academy of Sciences administers, edits, and has its members be involved in the review. It's a very good journal with a high reputation, and I think more importantly is widely read by influential people within science in the United States and around the world. So I pitched it to them as an opinion piece, and I think largely because Jeff is famous, that the PNAS office said, okay, we'll take a look, So we drafted an article where The first half, where the article was really asking for and pointing out the importance of an unbiased, apolitical inquiry into the origins of COVID. And the second part was some new and additional information extending some of what had been written following the revelations of the so-called DEFUSE grant proposal. So the DEFUSE grant proposal was concocted by a group including EcoHealth Alliance under Peter Daszak, the Wuhan Institute of Virology, and also the University of North Carolina at Chapel Hill with the group of Ralph Barrick. Many things were included in the DEFUSE proposal, but one of the things that was included was a proposal to insert human-specific protease sites into viruses that were being discovered, novel coronaviruses that were being discovered through the prospecting activities of EcoHealth and Wuhan Institute of Virology. This was particularly important because we all know now that SARS-CoV-2 alone, among hundreds and hundreds of members of the group of sarbecoviruses, is the only one that has this furin cleavage site insertion at the S1, S2 boundary. Not only is this unusual, but it's incredibly important for the pathogenesis of the virus. It makes it much easier for the virus to infect cells. And experimental virologists have shown since that removing the furin cleavage site from SARS-CoV-2 reduces its ability to infect human cells and also reduces its transmissibility between animals in animal models.
So you, Jeffrey Sachs, published that paper in 2022. It causes a bit of a kerfuffle. You then have been tracking it ever since, even worked with congressional investigators on figuring out the origin of the virus. The reason I'm saying this is you have a very deep understanding both of the political situation and of the scientific situation related to discussions of the origin of the virus. Even though you've been out of the public view, you're not on Twitter. I don't think you've published anything since 2022 on the origin, correct?
Correct.
You have recently been featured in a film discussing the origin, as you know, that we're not going to talk about right now. But in fact, you're wearing the very same yellow hoodie that you appear on in that film, and it's Thank You, Dr. Fauci's the name of it. But what we're going to do, what I think will be helpful here, is for you to discuss the science of the COVID-19 pandemic.
So generally speaking, we think of epidemiology as being science. When we're talking about public health, then public health always has a political dimension because public health involves scientists and public health officials interacting with local politicians. And I think we've seen many examples where political imperatives and political factors have a profound influence on the activities and pronouncements of public health people. When we're talking about origins of COVID in Wuhan, one of the things I want to say is that science is not really a consensus activity. There are many examples in the past where a consensus of scientists has held a view that has subsequently been shown not to be true. Evolutionary biology is another of those fields that often doesn't generate testable hypotheses. All of the data is essentially historical, and it's not generally possible to generate new data. So the current model is that two different forms of the SARS-CoV-2 virus were transmitted from animals to humans in a time period that they're defining as being around the beginning of December 2019, and in a location that they define as the Huanan Market.
I understand I can't pronounce words, and you're shaming me for my inability to pronounce words, Neil. So we have two things that they're claiming based on, you can read these papers, but they're saying outbreak began, is it early or late, just sometime in December at the, I'll put your voice over mine, at the
Huanan Market
in Wuhan, China. Just as an aside, and maybe we'll come back to how absurd this is, they also say that Eddie Holmes took a photo of the precise point where the outbreak began in 2014. I just don't understand how this is so embarrassing, Neil. But okay, so there's a photograph taken in 2014 where the raccoon dog that started the pandemic by transmitting multiple strains was sitting in late December or whatever. So we have time, December, location,
Huanan
Seafood Market,
and raccoon dog.
Well, we're going with the record that we're not going to even talk about where it came from. We're just going to talk about where it started and what time, because that's going to be easy. So how did they reach the conclusion that it started just at the wet market, not in the raccoon dog cage, but just at the wet market?
So we'll back up a little bit. In December 2019, obviously people in Wuhan and people in the Chinese CDC were aware that something was happening in Wuhan. Patients were showing up in the hospital, and for a little while, it seemed as though those patients might all have a connection to this location, the seafood market, the Huanan market. For those who don't know, Wuhan is sort of divided by the Yangtze River, and on one side of the river you have the older part of the city, which has a somewhat older population, and also this is where the Huanan Seafood Market is located.
For the visual, since we're doing this in person, you could consider this the river, and then that would be the older side, and the younger side would be over here, if you want to do it that way. It might be helpful for people to orient them. So that you would be the
older side.
Seafood Market, and I'm whatever's over here, the younger side, I'm over here, if that's helpful. It might be helpful. I don't know.
Right. So the more modern part of the city is called Wuchang, and the older part of the city is called Hankou. In the older part of the city, the Hankou section, that's where the seafood market is located. Early on, the Chinese CDC people thought that there might be a strong association between the patients that they were seeing and the seafood market, but very quickly people in the Chinese CDC realized that there were patients in other parts of the city. So if you look at the timeline, some of the early December cases had either been to the wet market, or a member of their family had been to the seafood market, or somebody worked at the seafood market. But likewise, there were other cases that occurred during a similar time period that did not have connections to the market. That raised the possibility that there was already human-to-human transmission, which it took the Chinese quite a long time to actually announce formally that there was human-to-human transmission. Mm-hmm. They were hoping that it was simply animal-to-human transmission, and therefore that it could only have come from the seafood market. George Gao, the head of the Chinese CDC, admitted to people in the United States with whom he spoke, admitted by January that there was human-to-human transmission, and that it was not a case of all of these patients had connections to the seafood market.
So, okay, there's a couple things. Number one, it was thought, based on reasonable conclusions, based on available evidence, that the virus did not begin at the wet market, but somehow we now are in the day, like 4.5, 5 years later, where the scientific community is wanting to convince the public that it started in the wet market with these two jumps, which it's too complicated to even get into that, in late December.
Early December.
Early December. Okay.
Yeah.
So it really is odd. And I think it's weird because when I say this or you say this to a normal person, they're like, that doesn't make any sense. Why would people need to jump through hoops and, all this stuff to like deny what very early on was clear? So Neil, both you and I believe that there was a lot of people trying to cover stuff up. Just to be on the same page, in our universe, we believe there was a cover-up. The basis for saying it begins in the wet market is to try to point the finger to the wet market where there are animals, where transmission has occurred, like from animals to humans before, and away from the lab. Can we start there, and then you can explain why the conclusion that it started at the wet market is I don't even know what word to use, but it's unsound. How about that? Fair to say unsound.
So I think the phrase you're looking for is plausible deniability. So I think it's quite revealing that it has mainly not been Chinese scientists who've been pushing the wet market idea. And in fact, George Gao has said repeatedly that he doesn't believe that the outbreak in Wuhan began at the wet market. it's been mainly Western scientists. Let's sort of look at why is this view that the virus came from the wet market so prevalent, and why is it being pushed so hard? In the previous outbreak of the SARS-CoV-1, so the earlier virus that emerged in 2002 and 2003, the outbreak showed many differences from SARS-CoV-2. One of the differences was that there seemed to be a relatively slow adaptation of that virus to infect humans, whereas SARS-CoV-2 seemed to be quite good at infecting humans from its earliest detection. The other thing was that it's very quickly possible to identify links between the SARS-CoV-1 virus and a specific population of animals. In this case, it was the palm civet was identified as a potential host, but they were actually able to gather strong scientific evidence. So they were able to find infected animals, they were able to find infected meat, they were able to find people who worked in restaurants who'd handled the animals and who'd handled the meat who had signs of infection. And so the link between the animal source and the transmission from animals to humans had a scientific basis. That scientific basis is lacking in the case of SARS-CoV-2. In the case of the wet market, there's no evidence for infected animals at the market. What we have is evidence for virus at the market, but this is the exact same virus that was circulating in the city of Wuhan during a very large-scale epidemic. That virus was presumably brought to the market by the people who worked at the market and visited the market every day. So there's this hypothesis that the Wuhan market was the center of the epidemic in Wuhan and that it was responsible for the spread of the virus to other parts of the city.
So as a scientist, how would you test that prediction?
So the approach that was taken by one of the groups that was publishing in 2022 was to obtain the home addresses of the patients who had been admitted to hospitals with a disease that was diagnosed as being COVID-19. So at this point, they didn't necessarily have a PCR test, but it was a lung CT scan or X-rays that would reveal characteristic changes in the lung that are associated with COVID-19, severe COVID-19 disease. So they gathered the home addresses of all of those patients. They looked at the locations that those people lived in, and they drew some circles around where those dots were clustered together.
Okay, so you have dots on the homes. So you got your dots on the locations of patients from hospital.
Yep.
Okay, so now I've got a thing of dots.
Yeah. So we've got a field of dots.
Dots.
And then what they would do is they would go to the seafood market and they would put a circle around the seafood market and see how many of those dots
Wait a second.
they could catch.
Hold on. They say, okay, let's hypothesize that it began at the seafood market.
Right.
And now I'm going to start drawing circles out from there. Now, what's the control there? Do they put it down?
Yes. So this is so one of the problems that I have with the work of these groups is that there are a lot of missing controls. So controls are things that scientists do as a sort of counter-argument. So if my argument is correct, then this should also be true, but on the other hand, this should not be true. So, for example, if you were to draw a circle around the main Hankow railway station, which is actually very close, it's maybe 800 yards away from the seafood market, you also catch a lot of these dots.
But they're saying that you catch more dots if you start drawing circles from the market, or is that not?
They said
they must have tested a bunch of other places.
They didn't test very many alternative arguments, but they said that the seafood market was inside circles that contained many of the dots. So let's just say that this dot and circle
but
dot and circles.
I know, but let's make it clear that the way you do dot and circling is you have dots. Like you first put dots, but then you pick where you're starting your circles. You get to choose.
Yes.
You're not taking those dots and having it figure out where the circle should start.
Right.
You're putting the circle starts at specific locations you choose.
So in other words, predetermined circles were examined for their relationship to the dots.
To dots. And the dots are the experimental...
What Bryce is saying is that another way of doing this is to have your dots and then see what circles those dots would generate.
Whatever you're using.
And you could actually get those dots to draw circles around different groups of housing. There's a lot of senior housing there in that location. Senior people tend to get sick at a higher frequency than younger people. You could draw a circle around the Hankow railway station and it would have lots of dots in it.
But you'd have to say, okay, so it's capturing the dots is what you're doing. So the circles are going out and how many dots are inside the circles is the metric.
Let's assume that Bryce and I, we don't know what we're talking about, and that these people...
It's not an assumption.
Let's say that these people are the experts.
We don't have to make an assumption. I think you don't have to make an assumption. They used a model on Wuhan to draw the conclusion, right, that you get the wet market as the place where things started. If that model is good, we could apply that elsewhere, correct?
So let's say that this is the greatest model. The greatest that has ever been devised.
It could be.
So if that's true, then it should be able to describe what happened in other epidemics. An epidemic is a very large-scale but locally constrained outbreak. Pandemic is when an epidemic spreads from one location to multiple locations around the world.
Right. Now, if things spread to other locations in the world, in our modern society, they would enter through airports, correct? Maybe?
But let's make no assumption.
Oh, no. All right.
Let's make no assumption.
Okay.
So what other epidemics do we know about in large metropolitan areas that are comparable to the one in Wuhan? So we're here in a secret, undisclosed location.
Right. Please don't.
Yeah. But not too far away.
I don't want the drone strikes to take this podcast out.
Not too far away, there is a major metropolitan area.
Very far away. This could be anywhere from New Jersey to California. We don't know.
So in the United States, there's a major metropolitan area of around 10 million people that experienced a very large epidemic in 2020. The New York City epidemic of COVID-19 was very, very well characterized. We have a lot of data. It's very well described. And I was living and working in the middle of it. And so
we have patients.
We know their home addresses. We know their movements between...
We don't need movement.
Between home and work. Dots don't move.
Dots don't move. We can just use the dots.
Just use the dots.
Let's use
just the dots. Let's keep it simple.
This model, I've been told this model is
the best.
The best. And that if you disagree with the conclusions of it, that you are a lot of horrible things, which I am, but for reasons other than my disagreement with the dot model. But go ahead, please.
So we'll ignore Bryce and we'll just use the dot model because the dot model is so
tried and true. I mean, it's the dot model.
It's the dot model. Okay.
Dot and circle model.
So if we look at New York City at the early cases, we know where the early hospitalizations occurred, and we know where those people lived. The very first patient to be hospitalized in the New York City metropolitan area was a gentleman who worked in a law office across the street from Grand Central Station. He lived in the suburb of New Rochelle in Westchester County, and he commuted every day, which meant his dot was moving.
Yeah, but the dots...
No, dots can't move.
The model we're using is not a moving dot model. We're using it's a stationary dot model.
So he became infected, and he went to a local hospital in Westchester County, and then he was transferred to New York Presbyterian Hospital in Manhattan. And he became the first hospitalized patient within New York City. But his home address was in New Rochelle. Public health officials went out to Westchester County. They went to New Rochelle. It's an easy train ride. They could just jump on the train with their tests. Right? And they got to New Rochelle and they started testing people, and they found that lots of people who attended the Young Israel Synagogue in New Rochelle, along with this patient, had become infected with SARS-CoV-2. Not many of them had become ill, but many people had been infected. Many of his friends and family had been infected, and they were able to develop a map of dots, actually.
Dots, right.
Dots. So New Rochelle was suddenly...
But those are and again, those are the dots that are the earliest cases in New York, right? Like these are very early dots.
So these were associated with the very first hospitalized case in New York.
Okay, so now we have
so we got a bunch of dots out there, just Westchester County.
Is it fair to say that those dots would be comparable to dots in Wuhan, the dots we have in Wuhan? That's the question. We can't apply a model if the dots are not very, like the dots need to be...
No, we got a bunch of dots, Bryce. We got a bunch of dots.
We're not throwing dots out.
Okay, we're not throwing dots out. There's a bunch of dots out there in New Rochelle in Westchester County. Okay.
Okay, so let's apply the model.
Nope, not quite ready. Not quite ready.
I'm excited about applying the model.
Yeah, I'm excited too. So then there are, within a few days, other clusters of hospitalized patients that appear in New York City in different locations. There was a few in the Bronx, and those patients were admitted to one of the closest hospitals, which I think was St. Barnabas Hospital. So you've got a central Bronx bunch of dots now.
Okay, but those are later dots.
A little bit later.
Little later.
Not much later.
So we can because we can separate dots by appearance and location, right? So sorry, time of appearance and location.
So we got Westchester dots, we got Bronx dots.
Yes.
And then there were some in the neighborhood of Borough Park in Brooklyn. There were a bunch of dots, and those patients were admitted to Maimonides Medical Center in Brooklyn. And
Quick question.
Yeah.
Where can people find the dot? Like, where is this data? Is the data actually available publicly that somebody could access the dots of the homes? And could somebody run the model if we're unable to right here?
New York Department of Public Health.
Okay, so we could get the data from the New York Department of Public Health.
Yeah.
And okay, so that's available. There's a record of this. So we have now 3 clusters of dots.
Right. And then not long after that, a very large cluster of hospitalizations started to appear in Queens, in northern Queens, in the neighborhoods that are called Elmhurst and Corona. So there was...
That's very interesting. So you're saying that in a neighborhood called Corona,
there was an outbreak of coronavirus.
Which I guess is bound to happen if you name a town Corona, and there's going to be a coronavirus outbreak. But.
Yeah. Now, if we look at the dot plot...
You got your dots.
The dot map.
Stationary dot.
So we got dot maps. We got 4 different groups of dots.
You got 4 different... Yeah, let's apply... So are we going to apply the dot method to all groups or just individual groups?
So here's what you can do. You can make the dots draw the circle.
Wait a second. No, I'm so sorry.
No,
you can't... No, you don't do that. You pick where your circle starts.
All right.
So where would we like the pandemic to start, the epidemic to start in New York?
Let's find a seafood market.
Is there there are wet markets in New York?
Let's find a seafood market.
Wait, is there a raccoon?
There's no raccoon dogs.
White-tailed deer. There might
even on Canal Street, I've never seen a raccoon dog.
Okay, so there isn't a place where we can pin the epidemic. Now, actually, I would say that it came from if it comes into New York from outside, it would come into the airport, right? That'd be reasonable. Or by sea, one of the ports or something. I don't know.
Okay, so we'll backtrack to some real science now.
What we're doing is in real science?
I don't think so. I'm so hurt. I don't think the dots are real science.
Well, wait, we haven't proven they're not. I mean, so you're saying if you apply his math, well, we don't even know how to apply it because it depends. Wait a second. Hold on a second, Neil. Are you saying that I already assumed the conclusion before applying the model? So that doesn't seem too scientific, then.
So the reason we're talking about the New York City epidemic is because there is so much information. There is a very, very detailed study on the molecular epidemiology. In other words, looking at the sequence of every virus that they were able to isolate from different patients across New York City. Mm-hmm. And the team at Mount Sinai accumulated all of this data, collected all of this data, and analyzed it in great detail. This was published in a paper. The lead authors were Ana Gonzalez and Matt Hernandez, and they published this, I think, in September 2020. And what they were able to show is that there was multiple introductions of the virus into New York City. Most of them came from Europe. So the type of virus that was introduced into New York City was largely identical with ones that were in circulation in places like Italy. Obviously, there's a lot of travel between Italy and New York City. Many of those viruses seem to originate in Italy or in other parts of Europe. There was at least one case of the virus being introduced from Iran by somebody who returned from Iran, having spent some time there, during a period where the SARS-CoV-2 virus was in circulation in that country, because they were also experiencing a large epidemic that actually predated the one in New York City. More interestingly, the Mount Sinai group was able to show that some of the viruses that were introduced into New York City came from other parts of the United States. And specifically, they found the same virus introduced into New York City that was circulating in January and February in Washington state.
Can I just say with confidence that whatever argument is being used to claim that the outbreak began at the Huanan, did I say that right? Seafood / wet, the wet seafood market, whatever we're calling it, is using an approach. I'm not even going to call it a model, because it doesn't seem like it actually warrants being called a model, because it's just drawing circles and counting dots, which, to be fair, sounds like a grade school exercise. So how about this? If the dots define the circles, there's a model that we need. But we're saying that you pick a center for circles and draw circles and count dots within the circles you draw. And that way of approaching finding where something began is, I don't know what you'd want to call it. Should we just say it's not a good approach? It doesn't give you accurate information? Or is it just is it actually unscientific? I mean,
how about we call it bullshit?
Fair enough.
Okay.
Yeah.
So okay. We've just established that we've got 4 groups of dots in New York City.
Yes.
We've got one in New Rochelle. We've got one in the Bronx. We've got one in Brooklyn. And we got one in northern Queens. Now, you could draw as many circles as you like around each of those sets of dots, but none of those have any relationship to the way that the virus was actually introduced into New York City, because we know the answer. The virus came in from the airport. It came in at JFK, and it came in at LaGuardia.
That for sure. Assuming. Okay, the most plausible way you get hold on. Let's not now.
Newark. Let's not forget Newark.
There is a BSL-3, I think, somewhere close to here. But, I'm sorry. So you're saying that if the virus... So we've got one of 2 possibilities. One, Ralph Barrick's running a lab in New Rochelle, New York, which I don't think is true. We don't see any evidence that there is a coronavirus lab in New Rochelle, New York, but we can't exclude it. Or somebody came over in a plane and then got off the plane and moved. So the dots moving is something that wasn't factored into this dot and circle.
Just imagine the dots moving.
So to be serious here, Neil, this is egregious. If what you're saying, because you're the scientist here, is true, then this is so embarrassing for the virology community to be hanging their hat on it. So I would challenge Michael Worobey, right? He's the one that came up with this circle and dot method, to apply the circle and dots to New York City and see what he gets. Now, he might get the airport. I don't know. We can see. Let's have it out. Michael Worobey, please apply your model.
Challenge.
The Michael Worobey Circle Dot Challenge. If he would like, we can do a podcast, a Twitter space, where you apply your model. If you're so confident in your model, it's hard for me to be serious because I'm actually quite angry about this. I mean, how many people have died?
20 million.
Yeah. And so this is like what the scientific community is giving to the people that died, like framing it as this ridiculously stupid model, embarrassingly stupid. Michael Worobey and those with their names on that paper should be ashamed. If what you're saying is true, maybe this is an oversimplification. Maybe we have something wrong. But if you're describing to me that they have dots that don't move, even though they're people. Now, if the dots are dead, they won't move, but they're alive dots, right? And now we put circles in a specific location, and that's the basis for saying it's from a wet market and not from a lab. If that's true, then I would say that conclusion is unsound.
So I think we've established that it's quite difficult if you don't already know the location from which the virus emanates. I think we just showed that it's quite difficult, even with outstanding data for New York City, absolutely outstanding data in terms of quantity and quality. If we did not already know that it came from the airport There is nothing in the epidemiological data that would lead us to that conclusion, at least using the dots and circles method.
Is there a method? Does somebody come up with a method you have to apply your method in multiple epidemics? I mean, again, any approach or any technique that scientists develop, the test of the technique is, can it be applied to different situations and give you useful solutions?
So I'd invite the same group to take advantage. There's a fairly large database for Milan and Lombardy. So the smaller cities around Milan experienced a very substantial epidemic of COVID-19. There is a lot of high-quality molecular epidemiological data available, much of which has already been published by Italian scientists. And I would be interested to see how well the dots and circles method works there. Because again, one would suspect, I think quite strongly, that the international airport in Milan was the point of arrival for the virus. So I think we've beaten that one to death.
So in the first part of this discussion, we talked about how putting the location of the outbreak in Wuhan to the Huanan Seafood Market is based upon, I don't even know what we were referring to it. There's bullshit. I think you used the term bullshit, and it's very rare for you to curse. You cursing means a lot. So if the location is bullshit, is the timing also bullshit? As we talked about before, we believe there's a cover-up of the origin to draw attention away from it potentially happening at a lab. So it makes sense to point the finger at the Huanan Seafood Market to draw attention away from the lab. You called it plausible deniability. It didn't happen at the lab because if Mike Worobey, is that how you pronounce his name, or Worobey or Worby, or if he uses this completely unsophisticated way of putting dots on paper and then drawing circles out of the wet market, you can come up with a science paper that says that this thing started at the wet market. Now, the other piece of this is that they want to say that it didn't just start at the wet market, but it started at a particular time, December. Now, I get why you would conceal the location. If there is a cover-up, let's assume there is a cover. So the conversation's going to be bounded by both of us agreeing in advance that we're going to be given that people are trying to deceive about the origin of COVID. So we just have to admit that right up front. We get why you're trying to point fingers away from the lab. Why would you want to point fingers away from the correct timing of the pandemic, assuming that December is not the correct timing?
There are two possible answers to this. One is if the time and the location have to be fixed together. So one of the criticisms that was made of the wet market as the origin was related to the fact that essentially all of the virus that was found at the seafood market was the so-called B lineage. Now, most molecular epidemiology people agree that the A lineage, which differs, I think, only by 2 bases from the B lineage, and this includes really outstanding people like, for example, Jesse Bloom, who's a Howard Hughes investigator. So he's an outstanding scientist who doesn't always agree with these groups. He has pointed out, as have many others, that the A lineage of the virus is thought to have arisen earlier than the B virus.
Well, I mean, there's universal agreement, right, amongst the virologists, the domain experts, that A preceded B. Is that correct?
I believe that is the case.
Okay, so the problem you run into is that there's more B than A. There's only one sample that was A?
So at the wet market, so remember the sampling was not done by these Western groups that have published, but it was done by the Chinese CDC under George Gao, who was then the head of Chinese CDC. And they sampled extensively at the seafood market. They found essentially only the B virus. The A virus was found on one single sample associated with the glove. Most scientists, I think most reasonable scientists, would suspect that a single sample might be contamination. So there isn't really strong evidence of A being at the seafood market ever.
Well, but let's assume that's legit data. Right. Let's assume that it is. So A's on a glove. And...
Yeah, but most of it's B. So that already...
Well, how many samples are we talking about? Like we got 100.
Hundreds.
Okay, so we've got hundreds of B.
Yeah.
and 1A, and the problem is that A came before B.
Right. So this tends to suggest to, I think, reasonable people and reasonable scientists that the seafood market was not an early event, that it was a super spreader event that occurred later in the Wuhan epidemic, and that is supported by the apparent absence of the A virus.
When were these samples collected?
These were collected early in January.
So you have the cases of people that got sick. Those are being used, the dots and circles, as we talked about.
The dots are from...
Say it's coming from the market. But now you got a problem because now you have the viral samples from the market.
Are all B. Right.
Well, no. Yeah. One is A. One is A. One is A and hundreds are B.
Yeah. So that physical and molecular evidence is consistent with the seafood market being a super spreader event and not an early event. Now, I think it's worth recalling that clusters of hospitalizations during epidemics and super spreader events during epidemics are not usually early. They are either in the middle of the epidemic or they can occur quite late.
By the way, can I point something out?
Yeah.
Even before we get into this, I'm embarrassed. Again, we're really saying there's 1A, hundreds of Bs. Do we actually need to go into further detail? Most scientists, if they heard this, would say...
Well, let's do a little roll call. So the people who don't believe this story, George Gao, the guy who led the group that gathered the data.
George Gao, CDC director of China, believes that this is not
his equivalent at the time in the United States, Robert Redfield.
Also a member of biosafety now.
Does not believe this story.
Okay.
Ralph Baric, the world's preeminent coronavirus engineer, does not believe this story.
Who some suspect had some involvement in either the design or the something of the potential laboratory virus if it came from a lab. So even he is would be too embarrassed to sign his name to this, correct?
Right. This is not...
Hold on. Yeah. But we've got both CDC directors in China and the U.S., and then the guy who is considered the world's coronavirus expert saying that the timing this outbreak to December is something that they think is
not
incorrect.
Scientifically sound.
Not scientifically sound. Okay.
So let's do timing. So I'm going to do the same trick that we did for location. So we're going to talk about the New York City epidemic again. And again, we're going to take advantage of this fabulous data set that was collected and collated and analyzed in great detail by the group at Mount Sinai.
By the way, that data is out there. Like we said, the Worobey with his circle and dot model.
Yep.
I have a suggestion / challenge. Could he also use... or whoever it is, Pekar, right? So anybody that's involved in this timing thing, could they just apply this to New York and see what they get? Or
they could, yeah.
Okay, well, let's put a pin in that and come back to it.
So since you mentioned Pekar, who's
first author of
the first author of one of the papers in 2022 about the wet market, was also the author of a paper in 2021. So I actually like to read. I
hold that against you, but
not only have I read both Pekar papers. So there's a Pekar 2021 as well as a Pekar 2022. And actually, they're quite different. But I actually went back even further. So there's a paper from 2020 that discusses the timing of the emergence of the virus in Wuhan. And that was actually published by two authors from London. So these are very well-respected evolutionary virologists, Lucy Van Dorp and Francois Balloux. It's interesting that there are early papers, and they're from 3 different groups, and they all point to October 2019 as an origin date. And I found that quite interesting because presumably these groups didn't talk to each other, and they all arrived at this similar conclusion. And these papers were all written in 2020 or 2021. So there's the Lucy Van Dorp and Francois Balloux paper, which points to October 2019. There is a paper by Pekar and Worobey and Joel Wertheim, which was published in 2021 in Science. And that paper pointed to October/early November. start date. And then there is another independent analysis from, I think it's Sudhir Kumar and colleagues, that again, not only points to an October 2019 origin date, but also proposes, as have others, including Jesse Bloom, that there was an earlier progenitor virus that preceded the A lineage and then the B lineage. So these are, I would say, rather detailed analyses. They're by 3 different independent groups.
So you're saying that 3 independent groups working on their own came up with a date of approximately October.
Which is interesting because it wasn't like nobody came up with April 2019. Nobody came up with June 2019. They're all October, rather consistent.
The methodologies that they use, were they actually using different methods?
So I think in evolutionary virology, I think that everyone has their own slightly proprietary methods that are distinct. Distinct. Now, I want to discuss the timing of the Wuhan epidemic.
Right.
We're going to also take advantage of that New York database.
Okay.
We'll start with New York. So the peak of the epidemic in New York City was April the 8th in terms of patient deaths. The peak of hospitalizations was a few days earlier. And the earliest hospitalization we know was just at the very end of February. However, the molecular epidemiology, and again, I go back to the 2 wonderful papers from the group at Mount Sinai. So the first paper by Ana Gonzalez shows multiple introductions into the New York City area from a variety of sources, many in Europe, one in Iran, and some in other parts of the United States. And they were able to time those introductions as far back as January. So they're already going 4 to 6 weeks earlier than the first reported hospitalization. But in a second paper, Matt Hernandez and Ana Gonzalez and the rest of their group revisited their data and, taking advantage of some of the knowledge that had been gained in terms of the extent of asymptomatic transmission, they were able to speculate that there may have been many introductions where transmission of the virus wasn't ongoing. In other words, that the chain of transmission died out. And so they postulated that it was completely feasible for there to have been earlier introductions that went undetected, so that introductions into the New York City area could have occurred as far back as early January or even earlier. That gives us a time delay between the first introduction, or the very first introduction into New York City, and the first hospitalization that could be as long as, say, 6 to 8 weeks. Okay, so we'll just keep that in mind, that period of 6 to 8 weeks, and we'll go back and talk about Wuhan. What we know about Wuhan is that the earliest hospitalizations that are now accepted by the local authorities and by Western groups who've analyzed the data are hospitalizations that occurred early in December. And these are some of those hospitalizations that generated the dots, that generated the circles.
No, the circles.
Right. The circles were there.
That would have been a good way to do it if you took the dot. Right.
That's right.
So the circles, you just picked where you wanted to start your circles.
Yeah.
So
if we take the Wuhan public health data at face value, in other words, those were genuinely the first hospitalizations, then you would have to go back 6 to 8 weeks from early December to get back to the origin infections, or the earliest introductions.
Okay.
And that takes you to early to mid-October. So this all makes perfect sense.
That's right. And not only does it make perfect sense, but it also was something that has been concluded by multiple people.
And again, I have to remind everybody that you can still find this as well. The South China Morning Post had an article about hospitalized patients as early as November 17th. You can
still track with this October.
You can still find that, right? And again, that's within a sort of 6-week interval from a putative first infection. So that would again take you back to early October.
And we're not even saying where this could have even come from. We're just saying that the timing,
Right.
the actual scientific analysis of available data puts us to October. And this is something that is agreed upon by Ralph Barrick, George Gao, Robert Redfield. And
Lots of serious people and us.
well, okay, right. I think you're more serious than me, but still, it doesn't seem to take a genius to see something as amiss here.
Right. So the question is, so we did say that we'd stay away from talking about motivations, but...
okay, well, I don't know.
No, we didn't. We didn't say that. So I think we've convinced ourselves and hopefully others that the idea that the outbreak in Wuhan began early in December at the wet market is wrong and wrong, okay? So the date has to be wrong. Epidemics don't just start like that. They grow very, very silently. They're undetected. The New York City epidemic, we were waiting and waiting and waiting. We knew it would happen, but it was completely silent until that first patient appeared at the end of February. But the epidemiology shows that the virus was being distributed around the city, probably largely by the subway system, throughout January and February. I actually stopped riding the subway in the middle of February because I was so convinced that this was going to happen, and I just walked to work and walked back. And of course, everyone thinks you're crazy when you do this.
Wait, who thinks you're crazy?
Everybody thinks.
The hoodie's not helping.
The hoodie fair. Is this... the hoodie's available at Biosafety now?
Not yet.
Much?
The colors, the color's special. So we got it's like a light, it's a Dijon
mustard. It's mustard.
Dijon mustard.
Coleman's
mustard, yes.
It's an English
Hopefully
thing.
they'll sponsor this podcast after this, but I don't know. It's probably too politically hot. So here's the thing.
Yeah.
This doesn't take any scientific knowledge to see. I hope, I mean, certainly that dots and circles thing, I think I could probably explain that to a sixth grader.
Grade schooler? Yep.
Maybe even, we should bring in sixth graders to see if they buy this argument.
Yeah.
The timing's a little bit different, but both of these things seem very, very obviously incorrect. And yet we sit here in late 2024 with the virology community and the journals trying to still stick with this story.
So into 2021, so the Pekar, you did some homework on this a few hours ago. So the Pekar 2021 paper was submitted when?
April 2021.
Right.
Pekar, yes.
So in April 2021, it was obviously okay to talk about the fact that the virus might have emerged in October.
Right. This is useful to know. Yeah.
Right.
So, okay.
This is important because the next group of papers that started appearing in 2022 were the ones where suddenly it had to be December.
Why did they have to move the timing somewhere between 2021 and 2022? What is it explained by? So so is there a reason why you'd have to move the timing?
So in September 2021, the DRASTIC group, Internet Sleuths, had revealed the existence of a grant proposal from EcoHealth Alliance, Wuhan Institute of Virology, and University of North Carolina at Chapel Hill, describing their proposal to insert furin cleavage sites into viruses that were part of the same group of viruses as SARS-CoV-2. And we know that to this day, no other virus in that group of viruses, the sarbecoviruses, has appeared that has the furin cleavage site. It is unique. No obvious precursors to the furin cleavage site exist in related viruses, and this has become a major source of contention between groups interested in a possible laboratory origin of SARS-CoV-2 and those who are committed to the virus having a natural origin.
Before the DEFUSE proposal came out, there was there were credible efforts to figure out when the virus emerged, including from Pekar or whatever, to put it, to be more accurate, right? And so they had it in October-ish. So up until this point, they were making arguments based on the genetics, right? The genetics of the virus. You could find the components of the virus in nature, so it must have come from nature until proven otherwise. Now DEFUSE comes out September 2021, and now there's problems. Until then, it was okay for the wet market to just be a place where there was a super spreader.
Yeah.
But now you see diffuse. So is this the potential idea that you've got diffuse presents a big problem?
Right.
So actually the arguments that came from nature are way more credible based on the genetics than the nonsense that follows, even though the point is they're saying that we see these elements in nature, so it has to come from nature, which is just ridiculous anyway. But the logical fallacies or the ridiculousness ramps up after diffuse. Is that fair to say?
A number of people have stated that when the diffuse proposal was unveiled, their perception of the possibility that the virus came from a lab increased by an order of magnitude. I, for example, was ready to consider two scenarios: natural origin or accidental lab-associated release due to people going to collect virus and bringing it back unaware of its pandemic potential. The idea that the virus might have been manipulated had been raised very early on, if you remember. There had been speculation about the furin cleavage site within the virology community. We know this because there was a paper published, I think this even came out before the infamous proximal origin paper. A paper was published in Emerging Microbes and Infections. And this EMI paper, which included as one of its authors Susan Weiss, who is a member of the National Academy of Sciences, this paper was preceded by a bunch of emails between the authors where they said, oh my God, this looks like it might have been engineered. And
specifically pointed to the...
And I would hate to think that the furin cleavage site was inserted. And actually, both Barrick and Zhengli Shi were contributors to that paper who were not acknowledged by...
They were asked for input and they provided input.
They were not listed in the acknowledgement, so they were not listed as authors. And that in itself is fraud.
Which apparently isn't a thing in virology, I will say. And we've called for the retraction of that paper and are waiting for the EMI ethics and integrity panel to weigh in.
Right. But even not regarding those emails, they managed to get the paper published 10 days later saying there is no evidence.
No credible evidence.
No credible evidence. So that came out, and then the infamous proximal origin paper doing the same thing, but by more famous people and in Nature Medicine came out very shortly afterwards. So the unique and unusual nature of the furin cleavage site had been noticed by everybody in virology very early on, but cold water had been thrown on that immediately to silence that discussion. That was a very successful operation, very successful PR operation, because discussion of the furin cleavage site disappeared until the diffuse proposal was unveiled by DRASTIC. And when that happened, and we all got to see that groups that were fully capable of identifying new novel sarbecoviruses and fully capable of inserting furin cleavage sites into those viruses have proposed to do just that.
In 2018, and the grant, which they say, well, wasn't funded, so we didn't do the work, which, of course, as scientists, we know is ridiculous because scientists are always doing work that they're proposing. It does suggest that now the alarm bells go off.
Right.
Because, and one other thing, like the diffuse proposal itself was not covered. The existence of it was not covered by the big media. And still to this day, a lot of people are unaware of the existence of it.
So it was only after diffuse and also around the same time as Jeff Sachs and I published our opinion piece in PNAS. It was about that time that the papers started to appear where the timing of the outbreak in Wuhan was shifted.
It's just amazing. If you think of the timing, you go from April 21, where there's this paper that puts it in October.
Yeah.
And now, less than a year later, they're putting it to December. This is the weird thing. We still don't understand why they have to move the timing. I get why they have to move
But then it wasn't just...
sorry, because you have to pin it on the wet market and those samples that they knew don't really work so well for it to be the origin.
Yeah.
Unless it's December. I don't even know. I can't even figure this out. When you're trying to figure out why people are covering stuff up, all I can say is the science here is shit. There's not science.
This was a very coordinated campaign about the origin and the date.
Mm-hmm.
And so I began to hear about the origin and the date from a group of people who I knew had not read any of the papers. So this group includes a few scientists who were sort of bystanders. In other words, they were vaguely interested in the story of where the virus came from, but they were not the kind of people who would actually read the papers and look at the data.
Right.
And it was also people, well-meaning and very nice people inside virology themselves, who also couldn't be bothered to actually read the papers and look at the data. So these are the kinds of people who signed the 87 people who signed the paper that went to the American Society of Virology journals.
Right. So, okay.
So they did a very clever trick. So they managed to publish the same paper 3 times in 3 different journals at the same time. So one of the things we've never talked about is that this loosely formed group has vastly improved their citation indices by not only by writing papers, some of which we think are actually junk, but then by writing other papers that cite their papers. It's become like this enormous cottage industry of self-citation amongst this group.
One of the things that we didn't mention was the letter in Science that was organized by Alina Chan and has been co-signed by Michael Worobey, Ralph Barrick.
Investigate the origin of SARS-CoV-2.
That's right. And that was published when? Before or after DEFUSE?
Before, I think. Ralph Barrick didn't want to do a lot of talking after DEFUSE.
I guess, you know what, let's not even impeach motives here. The papers that they put out, certainly after the DEFUSE proposal, are just horrific and embarrassing.
So just going back to raccoon dog very quickly.
Raccoon dog, okay.
The evidence for raccoon dogs being involved is based on the fact that they were able to find sequences that were part of mitochondrial DNA from raccoon dogs that was in George Gao's data set. So, in other words, some of the swabs that they had used at the market when the Chinese CDC went in there, some of those swabs came back with DNA sequence that reflected the existence of mitochondrial DNA from raccoon dogs. So, they found mitochondrial DNA from animals in the swabs from the market, and they found RNA for the virus.
Right.
The RNA for the virus corresponds exactly with the RNA of the viruses that were circulating in people in Wuhan. My analogy, so I love to argue by analogy, as you will have realized by now. My analogy for this is that if you were to go into, what do you guys have down here? Wegmans, Piggly Wiggly?
We do not have Piggly Wiggly.
Yeah, I love Piggly
We have Wegmans. Yeah, we got some Wegmans.
So let's say you went to the Wegmans, and you went to the Wegmans during the peak of the annual flu season,
okay.
and you took some swabs. Those swabs would come back with genetic material corresponding to the influenza virus that was
circulating in humans. Let me get,
yeah.
Okay, that makes sense.
Good.
Yeah.
Good. Go to the top of the class.
That means that it's the people who put that stuff there. People put it there. People
brought the influenza virus.
And then sneezed and touched stuff, didn't wash their hands, looked at the apples and stuff.
No,
don't buy the apples.
It's normal, right? Right.
I guess so.
Yeah. So what else would be on those swabs if you took swabs around the meat counter?
Meat.
You get okay.
Yeah. Meat, DNA.
So let's talk
the meat.
Let's talk about meat. What is meat?
What do you mean? What is meat?
If you asked a
slaughtered animals.
If you asked a biologist what is meat in terms of what tissue?
Muscle.
Muscle, good. Skeletal muscle, right?
I don't know. I'm not a butcher, man. Look,
skeletal muscle. Okay, you get meat is a combination of skeletal muscle, bone, and fat.
Okay,
right. If you buy,
look at me when you said fat.
If you buy a cut of meat, this is what you're getting. Okay. So what is skeletal muscle? So, skeletal muscle is something that contracts and it uses a lot of energy, and the energy to fire those contractions of the muscle fibers is provided by mitochondria. So it turns out that meat, because it's skeletal muscle, is absolutely laden with mitochondria.
Okay.
And mitochondrial DNA which is a small circular piece of DNA, is very stable.
Okay.
So if you go to Wegmans and you swab around the meat counter, you will come up with a lot of mitochondrial DNA. Okay.
Yes.
Now, that mitochondrial DNA can come from...
It could come from a live animal or a dead animal.
Okay. Now, let's go in the back. If we can go in the back at the butchers, at the Wegmans.
They don't have live animals.
By appointment, we can go in the back, and I guarantee that we won't see no cows. No cows.
That's true. That's true. I think that would... I agree with you here, even though we haven't done the experiment.
We'll see no cows.
No live cows.
No live cows. No live chickens.
No live chickens, no.
No. You might have some fish.
You will have live fish, yeah.
They could be alive.
Yeah. Yeah, you don't need to have live, but we do know that...
And definitely no live raccoon dogs.
Right, absolutely. No raccoon dogs, because I've never seen raccoon dogs.
But you will definitely...
During the worst of times here.
You will definitely have bovine mitochondrial DNA.
Right, so you don't need to have a living animal
just the
anywhere near anything.
Just the meat. Those swabs that they took at the Huanan Seafood Market.
That's correct.
Those can come back with the mitochondrial DNA of any animal whose parts have passed through the market.
Right, ever.
Because the mitochondrial DNA can sit around and actually be stable for long periods of time, several years.
But do we need the animal to be living to be the source of the outbreak?
We do not. Oh, to be the true source of the outbreak? Yes.
We do?
We do.
Are you sure? Because we can always post hoc come up with some other explanation.
Because we know that the virus itself, viruses of this type are not particularly hardy. They cannot live on surfaces.
You can't get transmission from butchered meat. You need to be living.
I believe that to be the case. So I was on an email with the New York Times, and I actually went through the argument about going to the supermarket. And then the conversation just stopped.
What do you mean it stopped?
So there was an email and there was some chatter. And then I said, look, if you went to the DRASTIC, which is a sort of gnarly supermarket in New York. No, it's very nice. It's very nice.
It's expensive. Oh, it's not a nice... Okay, well, anyway,
very nice. I don't want to criticize.
We hope they sponsor this podcast after this. Again, we are looking for a sponsor.
So if you went to the DRASTIC and you swabbed, you would definitely get some mitochondrial DNA.
Yes.
But that does not mean to say that...
You'd also get kicked out of the store if you're swabbing without any permission to go in.
That does not mean to say that you would find a cow in the back.
No, you would not. And so you put that in to the New York Times reporter, and then they lost your email.
They...
And that email conversation went away so they could publish that there's strong evidence that a raccoon dog started...
They'd already published it, and I was telling them that they were, their butt was,
that they had made a mistake.
Perhaps their head was...
They're providing what may not be super accurate information to their wonderful readership. So, Neil, taking all this together, does anybody care that this really makes a picture that
I would suggest that there are multiple constituencies that need to care about this. I think that virology and
I don't know. I said care, not need to care. Yeah, we can tell people they should care, but what would that do to their livelihood? Like, what would that make? So, like, if you've been thinking,
I'm talking to the American Society for Virology right now. I'm talking to you.
Oh boy, they're not watching this, but if they are,
and I'm
watching this,
I'm saying that you should care about this because eventually the low quality and poor behavior of people representing virology is going to have a very deleterious effect.
If and when there's ever a change in the administration from Democrat to Republican, that's when it will happen. Because at this point, it divides by politics. You don't think so?
No. Another group that needs to care about this is the National Academy of Sciences. The National Academy of Sciences needs to care about this because the behavior by people in the virology and evolutionary virology fields towards scientists in other disciplines has been appalling.
No, they don't care. Nobody cares about these things. Nobody cares about bad behavior if it is in service of a noble cause, which they believe this to be, which is to protect the integrity, the fake integrity of something. Neil, I just disagree fundamentally that the scientists will care until it actually gets to the point where the grants are going away. Then they will care.
So let's finish, perhaps.
Yeah, we have to wrap it up.
Let's finish by talking politics very briefly.
Yes.
So this issue has been politicized for a long time. And one of the points that Jeff Sachs and I tried to make in PNAS in May 2022 is that the politicization of the issue is inappropriate. And furthermore, I believe that a small number of individuals has benefited from the politicization and the polarization of the community on this issue because it's enabled them to hide their bad behavior. Peter Daszak is still on Twitter complaining about the activities of the Republican-led committee that investigated the activities of EcoHealth Alliance. That was the Oversight Committee and also the Special Committee on COVID-19. I think what people don't realize is that the committees in the House that we just mentioned, but also the Homeland Security and Governmental Affairs Committee in the Senate, which looked into these questions. Both of those committees began with a lot of polarization and the Republicans in both cases leading the investigation. But I know, having had a front row seat for some of this in the Senate, that over time, as the investigation proceeded and as the hearings revealed more and more information, that the two parties came closer and closer together. And if you go back and look very carefully at the hearings that involved people like Peter Daszak and Bob Gary and Christian Anderson, you will find that over time, the Democrats on those committees became more and more skeptical of those particular scientists and their behavior. The Senate became much more of a bipartisan investigation with Senator Peters, who was the chair, joining the ranking member on the minority side, Rand Paul, in really pushing the need to increase discussions of biosafety and to look further into potential mismanagement of funds by grantees of NIH and misadministration or maladministration by people at the NIH itself.
Here's the thing. I've been very public about being willing to work with whoever wants to work on the issues that I care about. Accountability for what happened with COVID and getting actually effective regulations to prevent another lab-generated pandemic. That being said, without any doubt, there will be a sea change in policies if Republicans are controlling the levers of power in the government. That's just where we are right now. That is a bad thing for the country in terms of having only one party being interested in safety to prevent another lab-generated pandemic and getting closer to the truth about what happened in the first one. So I think, and you probably as well, will be more than willing to work with whoever, regardless of political affiliation, because it really should be apolitical. I mean, we've been saying, it should be apolitical. It's totally been politicized. That's just the way the world is right now.
The point I wanted to make is that I think at this point, it's only the obvious rogues who are clinging to the idea that this is purely political.
Look, I hope it goes away. I would love if it does become a Republican-led administration, to try to get Democrats to finally talk. I mean, I've tried to talk to Peters and his own. I'd love to talk to Democrats, but and try to work with Democrats. If they lose a lot of power, maybe then I don't know. But anyway, I think
one of the signs that we're making progress on this is that there is a bipartisan bill.
Oh, absolutely.
That has already passed through the committee stage.
Yes, the Risky Research Review Act, which is something that both you and I have been supporting. And it was a very, very productive dialogue between Senator Peters and Senator Paul that came up with what is actually, it is a compromise bill, but it's a strong bill. The initial bill that was put out, there were some issues, rightfully so. People were pointing out that there were some issues with it. They worked very hard behind the scenes to come up with a compromise that I think overall is something that's great for everybody. And it addressed the valid concerns that were made by several people. Right now, there are some people that are still opposing it. Honestly, you know what? Who cares? These people, the people who oppose it now, they're not operating in good faith. I'll simply end it with that. Neil, thanks a lot for joining me today for this, what I hope is a useful dialogue on the science, quote unquote, behind the origin of COVID. So thank you everyone for joining us, and I guess we will see you. Will we see you again? I don't know. I'll see you again next time on Science from the Fringe.